Welcome back to Bookfulness. This is episode 8. And last time we really got into genetic drift, didn't we? That whole random side of evolution.
We did. Lots of focus on population size, the big N.
Yeah.
But today, today we're putting evolution on the ground, literally,
Right. We're moving from the sort of abstract math of genes in one place.
to how real geography, mountains, rivers, temperature, just plain distance, shapes how life changes, how it adapts, how it diverges across space.
Okay, let's unpack this. We're tackling chapter 8 from the source material today, evolution in space. So less about just the genes within one group.
And more about how different groups, different populations interact across the landscape. It's
Right.
about connection, or sometimes the lack of it.
So the mission for this deep dive is understanding how the physical world shapes genetic patterns.
Pretty much. How the environment, the actual place, drives local adaptation, and also how the movement between places affects everything.
Movement, like migration.
Exactly. Gene flow. It's fascinating because sometimes just how far apart two groups are can be way more important than, say, the specific predators they face.
Okay.
We're really looking at this constant evolutionary tug of war
Tug of war. Between what?
between the forces pulling populations apart, that's selection driving local adaptation, and also random drift, and the main force pulling them back together, trying to make them similar. That's gene flow.
Okay, selection and drift versus gene flow.
Nailing down how that balance works.
Yeah.
It explains so much. Why species live where they do, why some adaptations stick and others don't. Even stuff like disease resistance patterns. It's fundamental.
All right, let's get into it. Where do we start? Patterns first.
Let's start with the patterns. What do we actually see out there?
Okay, so if you look at a species that lives over a really big area, say a plant that grows all the way from Florida up to Maine, you probably wouldn't expect to find, short plants just suddenly stopping at the Virginia border and tall plants starting immediately after. It's usually smoother than that, right?
Exactly. What you almost always see is a gradual change.
Yep.
Maybe the plants get progressively taller the further north you go, or maybe their leaves get thicker. Or could be anything.
And that smooth change across the landscape,
Right.
that's the core idea here.
That's the starting point. Spatial variation. Traits, allele frequencies, they aren't just scattered randomly.
Yeah.
They often change in predictable ways as you move geographically.
Okay.
And the technical term for that smooth, gradual change in a trait, or maybe the frequency of a gene over a geographic distance, that's called a cline.
A cline. So it's
Yeah.
like a biological slope. Evolution responding to a geographical slope, like temperature or rainfall.
That's a great way to think about it. A biological gradient matching an environmental gradient, altitude, latitude, moisture, you name it.
The source material had a really cool example. It's something about clover and cyanide.
Ah, yes. The white clover, Trifolium repens. It's a fantastic, almost counterintuitive example of a cline driven by selection.
Counterintuitive how? Because it
Right.
involves the plant kind of hurting itself.
Precisely. It forces us to rethink what's always good for an organism. So, white clover normally makes cyanide. It's a chemical defense.
Against what? Things eating it.
Yeah, herbivores like snails or rabbits. Inside the clover cells, there are these little compartments, vacuoles. They hold two chemicals separately. When the leaf gets damaged, chewed on the compartments break, the chemicals mix, and boom, hydrogen cyanide. Nasty stuff. Tastes bad, potentially poisonous, great defense.
Okay, sounds pretty useful. So why isn't it everywhere? The cline part.
Right, you'd think making poison is always good.
Yeah.
But if you map out the frequency of the genes needed to make cyanide, specifically one called CYP79D15, you find something striking.
What's that?
The frequency of the working cyanide-producing version of that gene drops dramatically in colder regions. It lines up almost perfectly with the 0 degree C average January temperature line the isotherm.
Wait, 0 degrees Celsius, freezing. So in places where it freezes regularly, the clover doesn't make cyanide.
Largely, yes. Selection seems to be actively removing this defense in cold climates.
Why on earth would it do that? Trade poison defense for it. What? Surviving the cold seems like a weird trade-off.
It tells you which danger is worse, locally speaking. The hypothesis is really elegant. When the temperature drops below freezing, ice crystals form inside the plant cells.
Okay, like frost damage.
Exactly. And those ice crystals physically rupture those little vacuoles holding the cyanide precursor chemicals.
Uh-oh.
So the chemicals mix inside the plant's own undamaged tissues. The plant poisons itself. Self-destructs, basically.
Wow. So the plant that can make cyanide dies when it freezes hard, but the mutant one that lost the ability, It might get eaten more, but it survives the winter.
That seems to be the case. Survival through freezing is more important than defense against herbivores in that specific environment. The fitness calculation completely flips.
That's amazing. Local adaptation in action. The environment makes a defense mechanism a liability.
Exactly. And that maintains this really clear high cyanide frequency in warmer areas, low frequency where it freezes. It's selection overriding a generally useful trait.
And this idea of clines. It applies to bigger things too, right? Not just single genes, like body size.
Absolutely. A really classic example, one you might have heard of, is Bergmann's rule.
Oh, yeah, I think so. That's about animals getting bigger in colder places, like moose or bears.
That's the one. Generally, for endotherms, warm-blooded animals, populations in colder, higher latitude environments tend to have larger average body sizes than populations closer to the equator. Think polar bears versus, say, sun bears.
And the reason is just geometry.
Pretty much. It boils down to the surface area to volume ratio. Heat is produced by the body's volume, but lost through its surface area.
Okay.
As an animal gets bigger, its volume increases cubed, but its surface area only increases squared. So bigger animals have relatively less surface area for their volume.
Meaning they lose heat more slowly.
Exactly. They're more efficient at staying warm. So as you go north, the selective pressure to conserve heat increases, favoring larger bodies, creates a nice smooth cline in average size across the species range.
That's a huge scale, continents even. But you can get adaptation on tiny scales too, right? The book mentioned grass near a mine.
Ah, the cobber-tolerant grass. Agrostis capillaris. That's a prime example of local adaptation happening over incredibly short distances. Hyperlocal, you might say.
Copper mines. So toxic soil.
Very toxic. In places like Wales where mining occurred, the waste soil, the tailings, are full of heavy metals like copper, lethal to most plants.
But some grass grows there.
Yes. Populations growing right on the contaminated soil have evolved high tolerance to copper. It's a huge advantage, obviously, if you can survive where others can't.
But the amazing part was how sharp the boundary was.
Unbelievably sharp. Researchers took samples. Grass growing right on the toxic mine tailings, tolerant, literally a few meters off the tailings onto normal soil. The grass there is completely intolerant.
Wow. Just a few steps changes everything.
It shows just how incredibly strong selection can be. It's maintaining that tolerance allele at high frequency right on the toxic patch, despite pollen potentially blowing in from the non-tolerant plants just meters away.
Pollen blowing, and that brings us to the other side of the coin, doesn't it? The mixing.
Exactly. That copper grass is fighting an uphill battle against mixing. What happens when mixing gene flow is the dominant force, not the exception. That's where we need to go next.
Right, so that image of pollen drifting or animals wandering just a little way, that constant mixing is gene flow. And if selection and drift are pulling populations apart, making them distinct, gene flow is doing the opposite.
Precisely. Gene flow is the great homogenizer. It's simply the movement of alleles between populations. Happens when individuals disperse or their gametes like pollen or sperm move or seeds travel.
And it has sort of two main jobs in evolution.
Yeah, a really critical dual role. Let's take them one by one.
Okay, first, the homogenization you mentioned, making things similar.
Role 1, equalizing frequencies. Gene flow tends to make allele frequencies more similar across different populations. Imagine two ponds of fish. Pond A has 90% of a fast-swimming allele. Pond B only has 10%. If even a few fish move between the ponds each generation, over time, both ponds will start to converge towards some intermediate frequency. 50-50 maybe, depending on the migration rate and population sizes. It smooths out the differences created by drift or local selection.
Okay, it erases local quirks. What's the second role?
Role 2, introducing new alleles. This is vital for adaptation. Let's say a really beneficial mutation pops up in Pond A. How does it ever get to Pond B?
A fish carrying its homes over.
Exactly. Gene flow is the primary way, other than the incredibly unlikely event of the exact same mutation happening again, that new, potentially advantageous alleles can spread throughout a species. It connects the gene pools.
So it prevents populations from becoming totally isolated, genetically speaking.
Right. Keeps the species cohesive, in a way. Now, if we want to measure this mixing, we need to think about how the species lives. Are they in distinct patches, like islands or those coppergrass plots?
Or are they spread out more evenly, like deer in a forest?
Right. For those discrete populations, isolated patches, we use the migration rate, usually symbolized as $.
And what does that represent?
It's simply the fraction of individuals in the population you're looking at that arrive from somewhere else in that generation. So if M equals 1/116, it means 10% of the individuals are immigrants.
Okay. And the source material had an equation for this. Equation 8.1, how does that help us understand the impact?
Ah, yes, delta P, LMPMP. It looks simple, but it's really powerful.
Break it down for us. Delta P, that's the change in allele frequency.
Right. The change in how common an allele is, delta P, in your local population depends directly on two things. First, the migration rate, how much mixing is there?
Okay, higher mem, bigger change.
And second, the term BMPI. That's the difference in the alleles frequency between the migrants and the local population.
So if the immigrants are genetically very different from the locals, the change is bigger.
Exactly. If the migrants have the same allele frequency as the locals, P equals 0, and even if M's is high, there's no change. Delta P equals a nuller. Gene flow only changes things if the incoming genes are different.
Let's try an example. Say our little pond population has a bright color allele at $p$1.1, only 10%.
Okay.
But a big nearby river population has that same allele at PM on $0.8, 80%. And let's say migration is pretty high, M on $2.22. So 20% of the pond fish each generation come from the river.
Right, so we plug those numbers in. Delta P equals 0.2 times 0.8 equal 0.1.
From that's $2 times $0.7, new $0.7, which is 0.14.
Correct. So in just one generation, the allele frequency in the pond jumps from 0.1 up to new dollars 1 past plus a 0.14, a huge increase.
Wow. So if that keeps up, the pond population will quickly start looking genetically just like the river population.
Unless there's strong selection in the pond against that bright color allele fighting back against the gene flow. But yeah, HIAM is a powerful homogenizing force. It can swamp out local adaptation or rescue a population from inbreeding. Understanding IM is crucial.
Okay, that's for distinct patches. What about the deer in the forest, or maybe plankton in the ocean, things spread out continuously?
Right, for continuous populations, we can't easily define them. Instead, we use a concept called the migration variance, written as sigmum 22. Sigmum squared.
Variance, like how spread out things are.
Exactly. It measures the average square distance that genes move between generations. If you take the square root, sigmum, that gives you something easier to picture. It's roughly the average distance between where an individual was born and where its offspring are born.
So it's not just how far they wander, but how far they move and successfully reproduce.
That's the key distinction. Effective dispersal distance. It measures how quickly genes spread, like diffusion.
And measuring this sounds hard. The book mentioned tracking lizards.
Oh, incredibly hard. The Texelite spiny lizard study, Celloporus olivaceus. Classic example of painstaking fieldwork. They had to mark thousands of newly hatched lizards. Thousands. Wow. And then recapture them later, track them as adults, figure out where they established territories and reproduced. Only then could they calculate the variance in the distance between birth and reproduction sites.
What did they find? How far do these lizard genes move?
The square root, sigma mart, came out to be about 99 meters. So on average, genes in that population spread about 99 meters per generation.
Okay, so that 99 meters is like the characteristic speed of gene spread for them.
Exactly. It lets you model how fast a new allele might radiate outwards or how large a geographic area needs to be before drift can start causing significant differences between the ends. It quantifies that diffusion process.
So whether we use MMOS or sigma M squared, we're measuring the potential for mixing. But how do we measure the results? Like, how different have populations actually become? Is there a single number for that?
There is, and it's one of the most fundamental numbers in population genetics.
FST. Okay. What does it tell us?
FST measures the degree of genetic differentiation or divergence among populations. Technically, it looks at how much heterozygosity, genetic diversity within individuals, is reduced in the subpopulations compared to what you'd expect if they were all one big, randomly mating pool.
Okay. Is there an easier way to think about the scale?
Yeah, the scale is pretty intuitive. AFST ranges from 0 to 1.
0 means?
0 means absolutely no genetic difference between the populations. They're effectively identical, like they're all part of the same gene pool, gene flow is very high, or they haven't been separated long.
And 1 means?
1 means they are completely diverged. They are fixed for different alleles of the genes being looked at. They share no genetic variation at those spots. Maximum differentiation.
What's typical in nature?
Most values fall somewhere between, say, 0.05 and 0.25. Anything above 0.15 or 0.2 is usually considered pretty significant divergence.
Okay, so FFT gives us a snapshot of divergence. Does it relate to geography? Like, do populations further apart usually have higher FFT?
Absolutely. That pattern is called isolation by distance, and it's exactly what you'd predict.
Why? Just less mixing over long distances?
Precisely. Populations that are geographically far apart exchange fewer migrants. Their M, or effective migration rate, is lower. With less gene flow to keep them similar, genetic drift and any local selective pressures have more opportunity to cause them to diverge over time. So VST tends to increase as the distance between populations increases.
The book used humans as an example of this, right? Something about Africa.
Yes, it's a striking pattern globally.
Yeah.
Human populations in Africa generally have the highest levels of genetic diversity, heterozygosity. As you sample populations progressively further away from Africa, across Asia, Europe, into the Americas, heterozygosity tends to decrease steadily. And correspondingly, the FSD values between populations generally increase with geographic distance from Africa and from each other along those migration routes.
And that pattern tells a story.
It reflects our species' history. The out-of-Africa migrations involved what we call serial founder events. Small groups budded off, moved, and established new populations. Each time, they only carried a subset of the genetic diversity from their source population.
Like a genetic bottleneck with each step.
Exactly. Losing variation and increasing divergence through drift with each migration step. It's a textbook example of isolation by distance driven by history and geography.
So 5T-Selects can track history. But can it also pinpoint spots where selection is happening right now, overriding that background drift?
Yes. And this is where 5Ts becomes incredibly powerful for finding genes under local adaptation. Even if there's decent gene flow, if selection is strong enough in one place, it can cause extreme divergence at just that one gene.
Is this where the sticklebacks come in?
This is exactly where the famous three-spined sticklebacks, Gasterosteus aculeatus, come in. They're like a natural evolutionary experiment.
How so?
Well, the ancestral form lives in the ocean and is covered in bony plates, good armor against marine predators. But over the last 10,000 years or so, since the glaciers retreated, these marine sticklebacks have independently invaded thousands of freshwater lakes and streams.
Okay. And freshwater is different.
Very different. Fewer big fish predators, often less calcium available to build bone. In many freshwater environments, those heavy plates become a disadvantage costly to make. Maybe slower swimming, maybe more vulnerable to insect larva predators that grab spines. Selection often favors reducing the armor plating.
So different populations ended up with less armor independently.
Many, many times, yes. So researchers did what's called a genome scan. They sequenced DNA from marine fish and from several different independent freshwater populations that had evolved low plating.
And then calculated VSD?
All across the genome, thousands and thousands of genetic markers. They were looking for regions with unusually high VSD values between the marine and freshwater groups.
What did they expect to find?
They expected most of the genome to show some low background level of VST just due to random drift and the fact that gene flow isn't perfectly total between marine and freshwater.
But
They found specific spots, little peaks on the graph, where VST went through the roof, way, way higher than the background level.
And those peaks mean?
Those peaks are the genomic locations where natural selection has been acting incredibly strongly and consistently in the freshwater environments to drive divergence, despite ongoing gene flow from the marine population.
Get this. One of the absolute highest, most significant peaks was on their chromosome 4.
Okay, and what's there?
It landed smack dab on a gene called ETA.
And
ETA is known to be the major gene controlling the development of those bony plates.
wow. So the gene directly responsible for the trait under selection showed massive divergence, while nearby genes, presumably neutral, showed only background levels.
Exactly. It's incredibly strong evidence. It tells us that local selection for low plating was powerful enough to overcome gene flow at that specific spot in the genome, like finding a smoking gun for adaptation.
That technique scanning for VST outliers has totally changed how we find the genetic basis of adaptation in wild populations. It's super powerful.
Okay, that stickleback example is perfect because it really sets up this core conflict, doesn't it? This tug of war you mentioned earlier.
It really does. You have selection trying to make the freshwater fish different, low plates, adapting them locally.
But you also have gene flow, marine fish potentially swimming in or interbreeding, trying to pull them back towards the high-plated ancestral state, homogenizing them.
Exactly. And the outcome, whether the population adapts locally or not, depends entirely on which force is stronger in that specific situation.
Is there like a simple rule of thumb?
There is, kind of. You compare the strength of selection, which we often denote with is, against the rate of migration, M.
Is versus if.
If selection is much stronger than migration is bringing X strain, then local adaptation usually wins. The population adapts despite the gene flow.
Yeah.
Like the sticklebacks at the ETA gene.
Okay. And if it's the other way around?
If selection is weak compared to migration, then gene flow dominates. The locally advantageous allele can't increase, and the population's genetics will mostly just reflect the migrants. Local adaptation fails.
And when that happens, when migration overwhelms selection and stops adaptation, that's called gene swamping.
Precisely. Gene swamping is when the influx of alleles from outside, often alleles that are poorly adapted to the local conditions, is so high that it prevents the locally beneficial allele from increasing or even drives it out.
Like drowning out the good gene with a flood of average ones.
That's a good analogy. The adaptation gets swamped. The source material gives the example of mosquitoes, Culex pipiens on the island of Corsica, dealing with insecticide.
Ah, resistance evolution. Classic battleground.
Right. So insecticide resistance alleles obviously provide a huge survival advantage where insecticide is used. Strong selection, high as. But on Corsica, insecticide was apparently only used in relatively small, isolated patches. Mosquitoes, however, can fly quite far. They have high migration potential, high sigma, meaning high effective dim, into those small patches.
So lots of non-resistant mosquitoes were constantly flying into the treated areas from untreated areas nearby.
Exactly. The gene flow of susceptible alleles into the small treatment patches was so massive that it overwhelmed the strong selection within the patches.
So the resistance allele, even though it was highly favored right there, could never reach a high frequency. It kept getting diluted.
It kept getting swamped. The population couldn't effectively adapt to the insecticide in those specific locations because the homogenizing force of gene flow from the large surrounding susceptible population was just too strong relative to the size of the area under selection.
A spatial failure of adaptation. Fascinating. And this connects back to clines, right? The shape of the cline reflects this balance.
Perfectly. We talked about clines as patterns, but why does a cline have a particular width or steepness? It's a direct consequence of this tug of war.
Okay, how does that work? There was another equation, wasn't there, about cline width?
Yes, equation 8.4. It's an approximation, but really useful. The characteristic width of a cline, $, is roughly proportional to the square root of migration variance divided by selection strength. So $, is width, sigma squared is migration, kickoff is selection. Let's unpack that relationship.
Look at the ratio inside the square root. Sigma of dollar. Migration variance over selection strength.
If migration's really high compared to selection, that ratio is big.
Right. Big ratio, big square root, big $. The cline is wide and flat.
Meaning the transition zone between the adapted and non-adapted types is really spread out.
Exactly. Strong gene flow smears the genetic difference across a large geographic distance, diluting the effect of selection over a broad area.
Okay. Conversely, if selection is really strong compared to migration,
then the ratio sigma to errors is small. Small ratio, small square root, small $. The cline is narrow and steep.
Like the copper grass boundary, just a few meters wide. Precisely. Strong selection maintains a sharp genetic difference right at the environmental boundary, resisting the homogenizing effect of gene flow.
And this is cool. Scientists can turn this around, right? If they measure the cline width and estimate the migration, they can figure out how strong selection is.
Yes. That's the really clever application. If you can measure dollars in the field, how sharp is the transition, and you have an estimate for sigma 222 for that species, how far do genes move, you can rearrange that equation to solve for keys, the selection coefficient.
The cline becomes like an evolutionary speedometer telling you how hard selection is pushing.
Kinda, yeah. A selection meter. For instance, back to Culex mosquitoes, but this time in southern France where resistance did become widespread in treated areas. Scientists measure the steepness of the cline for resistance alleles at the boundary between treated vineyards and untreated areas. They measured $. They had estimates for mosquito dispersal, sigma and 22.
And when they crunched the number,
they calculated selection coefficients as values, as high as 0.33, favoring the resistance allele inside the treated zones.
0.33. That means resistant mosquitoes had a 33% survival or reproductive advantage. That's huge.
Massive selection. The steepness of the cline confirmed it. It showed selection was strong enough to maintain resistance despite gene flow from nearby susceptible populations.
This whole idea leads to something pretty profound about like the minimum size for things to adapt.
Absolutely. There's a limit to the spatial resolution of adaptation. Meaning if a patch of favorable habitat is too small, adaptation might just be impossible, no matter how beneficial the trait is within that patch.
Why? Because of gene swamping again.
Exactly. The critical patch size is related to that characteristic cline width this one stubbs. If your habitat patch is much smaller than the other gene flow from the surrounding, Different environment will constantly swamp out the locally favored allele. It just can't gain a foothold.
So breaking up habitats into really small fragments that could actually prevent species from adapting to local conditions within those fragments?
It's a major concern in conservation biology. Fragmentation doesn't just reduce population size. It can fundamentally undermine the process of adaptation if the fragments are below that critical $Serraler threshold.
Wow. Okay, so most clines we've talked about involve selection changing across space, cold versus warm, toxic versus normal. But the book mentioned tension zones. Those are different.
They are. Tension zones are clines that can be maintained even if the external environment is perfectly uniform across the zone. The tension comes from genetics itself.
How does that work?
It happens when heterozygotes individuals with one copy of each different genetic type have lower fitness than either homozygote, we call that underdominance, or heterozygote disadvantage.
So if you have two different chromosome arrangements, maybe, that meet, the hybrids are less fit.
Exactly. Imagine two populations that were isolated and maybe different chromosome fusions became common in each one. Then they expand and meet. Where they meet, they hybridize. But if those hybrid individuals, the heterozygotes, for the chromosome rearrangement, have problems during meiosis, maybe reduced fertility, they have lower fitness.
So selection acts against the hybrids right at the boundary zone.
Yes. Even if the environment is the same on both sides, if an individual with chromosome type A wanders into type B territory, it might mate, but its hybrid offspring are less fertile. Same if type B wanders into type A. This selection against hybrids actively prevents either type from invading the other's territory,
creating a stable boundary, a tension zone, just because the mix is bad.
That's the idea. The source uses the grasshopper Podisma pedestris in the Alps. Different nearby populations have different chromosome fusions fixed. Where they meet, the declines in chromosome frequency are incredibly steep, maybe only tens of meters wide.
And that steepness is maintained by the low fitness of the heterozygote grasshoppers born right there.
Correct. The constant selection weeding out the low fertility hybrids maintains that sharp genetic boundary, the tension zone, independent of any obvious environmental shift across that narrow strip. It requires really strong selection against heterozygotes to keep the zone that tight against the constant pressure of dispersal.
Okay, we've talked a lot about gene flow as this homogenizing force and how it interacts with selection, but gene flow only happens because individual organisms move. They disperse.
Right.
So let's zoom in on that. Why do they move? Dispersal seems risky. You leave a place, you might get eaten, you use energy. Why does the evolution of dispersal happen at all?
That's a great question. Despite the obvious costs, energy, predation risk, maybe landing in a worse spot, dispersal evolves frequently because the benefits can outweigh those costs. There are sort of three main drivers.
Okay, number one.
Number one, finding better or simply available habitat. It's huge for species living in temporary or patchy environments. Think dandelions.
Ah, the weeds. Their seeds fly everywhere with those little parachutes.
Exactly. Dandelions thrive in disturbed ground lawns, roadsides, new clearings. Those patches don't last forever. Other plants eventually take over.
So if a dandelion lineage just stayed put,
it would eventually get outcompeted when its patch disappeared. Selection strongly favored lineages that evolved mechanisms like that parachute pappus to send their seeds far and wide, maximizing the chance that at least some will land in a newly opened, suitable spot. It's a strategy for dealing with unpredictable environments.
Okay, finding new real estate. What's number two?
Number two, avoiding competition, especially with your own relatives.
Kin competition.
Yeah. If all offspring stay right next to their parents and siblings, they're all competing for the exact same limited resources: food, water, space. By dispersing even a short distance, an individual gets away from that intense local competition.
So even if moving is risky for the individual, it might increase the overall success of the family's genes because the ones who stay behind have less competition.
That's part of the thinking, yes. It can be favored by kin selection. Spreading out reduces the pressure on everyone.
Makes sense. And number three, a genetic reason.
Number three, avoiding inbreeding. In many species, mating with close relatives leads to inbreeding depression-reduced fitness because harmful recessive alleles get paired up.
Right.
Dispersal, moving away from your birthplace before you reproduce, is a very effective way to ensure you're more likely to mate with unrelated individuals. It maintains genetic diversity and avoids the negative consequences of inbreeding.
So find good spots, avoid competition, avoid inbreeding. Sounds like good reasons. But there's always a cost, right? You can't be good at dispersing and good at everything else.
Usually not. There are often trade-offs. Energy and resources are finite. If you invest heavily in dispersal machinery wings, strong legs, lots of stored energy for travel, that's energy you can't use for something else, like reproduction.
The book had an example. A tricket.
Bush cricket.
Rosel's bush cricket. Metrioptera roselli. A really neat example of a dispersal fecundity trade-off, visible as a polymorphism.
Polymorphism means different forms in the same population. Yes, most of these bush crickets have short, reduced wings. They can't fly, but they reproduce a lot. They're highly fecund.
Okay, the stay-at-home have lots of kids strategy.
Right, but a fraction of the population develops long, functional wings. They can fly, sometimes long distances, allowing them to colonize new areas or escape deteriorating conditions.
But the catch is
that building and maintaining those flight muscles requires a huge energy investment. That energy comes directly at the expense of egg production. The long-winged flying morphs lay significantly fewer eggs than the short-winged ones.
So there's a direct trade-off, mobility versus reproduction.
Exactly. Selection maintains both forms in the population because the optimal strategy depends on the local conditions. If your current patch is stable and good, staying put and reproducing short wings is best. If the patch is declining or crowded, the ability to fly away and find somewhere new, long wings, might be worth the reproductive cost.
Okay, so that's the individual's choice, or rather, the evolutionary outcome for dispersal. Now let's zoom out again, to the whole species. What determines the limits of a species' range? Why does a species live here, but not there?
Ah, the million-dollar question in biogeography. Why do species stop where they stop? It's usually a combination of factors.
The obvious ones first, like oceans.
Yeah, large-scale physical barriers are often the clearest limits. Oceans for terrestrial animals, continents for marine ones, major mountain ranges, deserts. Things they simply cannot cross or survive in.
But sometimes the boundary isn't so obvious, is it? The habitat might look okay just beyond the range edge.
True, and that's where genetics and other interactions come in. One possibility is simply a lack of the necessary genetic variation.
Meaning they just don't have the right genes to adapt to the conditions just outside their current range? Like if it gets slightly colder or drier?
Exactly. If the environment at the margin requires some novel adaptation, but the alleles for that adaptation don't exist in the population's gene pool, or are extremely rare, they simply can't make the leap. They hit an evolutionary dead end.
And this ties back to our main theme. Gene swamping can also limit ranges, right?
Hugely important factor.
Yeah.
This is where the dynamics within the species prevent expansion. Imagine the edge of the range. Conditions are tough there, suboptimal. Selection might weakly favor individuals with alleles that help them cope with those marginal conditions. But the center of the species range, where the habitat is optimal, usually holds a much larger population.
And that large central population is constantly sending migrants out to the edge, gene flow.
Right. And those migrants carry the alleles adapted to the good central conditions, not the tough marginal ones. This constant influx of mainstream genes can swamp out the locally advantageous edge alleles before they can increase significantly.
So the species' own success in the middle prevents it from adapting at the edges.
It can, yes.
Yeah.
The core population genetically pollutes the edge, preventing adaptation to the novel conditions required for range expansion. Again, it comes down to C dollars versus better. Is selection at the edge strong enough to overcome the homogenizing gene flow from the center? Often the answer is no, and the range boundary stays put.
We also can't forget other species, right? Biotic interactions.
Definitely. A species' range might end simply because it runs into a superior competitor, a really effective predator, or a disease organism that it can't handle in that new area. The biotic environment can be just as hard a barrier as a physical one.
The book ends this chapter with a really extreme example of range limitation. A salamander in Texas.
Ah, the Barton Springs salamander, Eurycia osorum. A really sobering case study.
Why sobering? Because its range is tiny.
Incredibly tiny. Its entire known natural distribution is limited to the underwater aquifer system feeding Barton Springs and a couple of nearby springs. Right within the city of Austin, Texas. We're talking a few square kilometers total.
Wow, just those springs. How did it get so restricted?
It's become highly specialized. It's neotenic, meaning it retains larval features as an adult, and it's fully aquatic. It cannot survive on land at all. So it's physically trapped within that specific underground water system. It can't disperse overland to colonize other springs, even if they were suitable.
So extreme physical limitation. What about gene flow within its tiny range?
Gene flow occurs via water movement within the aquifer. It likely prevents any significant local adaptation within that small system, keeping it relatively uniform. But the critical point is the lack of any potential for dispersal out of the system.
So its extreme specialization and geographic confinement make it incredibly vulnerable.
Extremely. If anything happens to the water quality or quantity in that specific aquifer system, pollution, overuse, changes due to climate, the species has nowhere else to go. It has no evolutionary resilience through dispersal. Its tiny range, enforced by its biology and the geography, is its ultimate Achilles heel.
Okay, so wrapping this up, here's where it gets really interesting, I think. We've basically put evolution on a map today. We've seen that the patterns we see out there, the clines, the sharp boundaries, the species ranges, they aren't just random distributions.
Not at all. They're the direct result of this ongoing, quantifiable dance between local selective pressures, trying to create differences, and gene flow, trying to smooth those differences out. It's a dynamic balance.
We saw it in the clover giving up its cyanide defense just to survive freezing.
And in the sticklebacks, where we could pinpoint the ETA gene using those phi-ST peaks, proving selection beat gene flow at that specific spot.
And we saw how we can even estimate the strength of selection just by looking at the width of a cline, using that Allers formula, relating it to migration.
Right, and understanding that balance, that Allers versus Darn battle, helps explain why adaptation sometimes fails, like the swamp mosquitoes on Corsica, or why it requires a minimum habitat patch size to even get started.
Ultimately, this spatial dimension dictates the limits of life, doesn't it? It defines how finely tuned adaptation can be and why some species are stuck in tiny places like the Barton Springs salamander.
While others can spread across continents, it all comes back to the interplay of adaptation, the physical landscape, and the movement of genes across it. Evolution doesn't happen in a vacuum. It happens in specific places connected by movement.
So we've seen how populations change and adapt in space, and how they diverge genetically because of distance and selection. But how does that divergence become permanent? How does one group actually split off and become a totally separate species that can't interbreed anymore?
Now you're asking the big one. That process, the origin of new species, is where we're headed next time.
We'll be diving into speciation, how reproductive isolation evolves, the different ways it can happen, from geographic separation to genetic incompatibilities. It's one of the cornerstone topics in all of evolution.
Absolutely. Looking forward to it.
Us too! Thanks for listening to Bookfulness. Join us for the next deep dive.